Well, it's very nice to be talking in this membrane trafficking seminars. And I will be talking about trafficking to the lysosomes and mainly about the role of the Hobbs complex therein. And let me see how I go to the next slide. I try to get to the next slides.
Try to use the arrows on. Okay, good. Perfect.
Yeah, I do the arrows inside now on the bottom. Okay, there we go. So lysosomes are very interesting organelles, as we just heard from Arnaud. But they're also very interesting in the sense of membrane trafficking, because in fact they are at the crossroad of 3 trafficking pathways. And I think the most well known of that is the endocytic pathway, which starts at the plasma membrane to early endosomes, late endosomes, and then to lysosomes. But lysosomes are also the last part of the autophagy pathway, which starts with the formation of a phagophore, an autophagosome, which can then fuse directly with a lysosome or to a late endosome and then the lysosome. And the third pathway is the biosynthetic pathway from the ER to the Golgi, either via the plasma membrane or directly from the Golgi to the endosomal system. And the endocytic pathway and the autophagy pathway, they bring cargo to the lysosomes to be degraded. And the biosynthetic pathway brings the machinery, the resident proteins that are needed to perform lysosomal function. So there are many lysosomal functions. We heard a few of them already. And we know them, of course, most by their function in degradation, which is done by the soluble hydrolases, which are in the lumen of the lysosome. But there are also a lot of membrane proteins in the limiting membrane of the lysosome, which are involved in the transport of ions and breakdown products made by the hydrolases from the lysosome into the cytosol, by which new proteins can be built. There are also resonance proteins in the limiting membrane that are required for general lysosomal function and also protection of the lysosomal membrane. There are many associated proteins that are required for membrane fusion, but also to make contacts of lysosomes, for example, ER or mitochondria. And very importantly, lysosomes are the organelles in the cells that harbor the mTOR signaling complex. And via the VATPase, the mTOR complex can sense the nutrient status in the lysosomes and by that initiate signaling pathways to the nucleus. So today I want to first talk a little bit how do we define the distinct compartments of the endolysosomal system, and I will mostly concentrate on the endocytic pathway to the lysosome. And then the biggest part will be what is the role of the Hobbs complex in lysosome biogenesis. So this was the picture I started with, and so I think most of you will be able to recognize the compartments I indicated here, the mitochondria with their beautiful structure, the Golgi complex, a little bit more difficult, but still very easy to recognize, and of course the nucleus. But it becomes a bit more difficult when we are going to look at the compartments of the endolysosomal pathway. For example, we here see the early endosomes, and they are characterized by a relatively empty lumen. And they're surrounded by many small vesicles. These can be endocytic vesicles coming directly from the plasma membrane, but it can also be recycling tubules that form from early endosomes to recycle proteins back to the plasma membrane, or TGN. You also see some small dark dots here. These are gold particles. They are associated with BSA, and we gave them to the cells to load the endocytic system. So these are the early endosomes. Then we have the late endosomes. They are a little bit more dense, a bit grayish. Here are they, you see them here and here, which you are I just have to put the thing away here, but here is the late endosome. There's a lot of intraluminal vesicles in the late endosomes, and they harbor the proteins that need to be degraded in the lysosomes. And then finally, in the lysosomes, these are HeLa cells, just well fed, and lysosomes are quite modest by appearance, maybe a little bit boring. They are blackish, grayish. Sometimes you don't see so much in them. Sometimes you see a little bit more, and that very much depends on the status of the cells, and also, of course, the cargo that they have absorbed already. So you see that the definition of these compartments is quite heterogeneous and not as clear as, for example, for a mitochondrion. So these are the morphological identifications, and what we can do is we can add some molecular information on top of that by doing immunoelectromicroscopy of specific proteins that we know are associated with defined stages of the endolysosomal system. For example, here we see labeling of the transferrin receptor, which is in the limiting membrane of an early endosome, and also the cycling tubules. Here we see labeling of LAMP1, which is present in late endosomes, depicted here, and also in lysosomes. And here we see labeling of cathepsin D, which is a lysosomal enzyme, and you see the gold particles, which are very abundant in the lysosome. So this is what we call the morphomolecular characterization. But of course, we also have the functional analysis of the endolysosomal system, and also the molecular analysis by proteomics, by all the biochemical approaches. And those pick up many more proteins and very complex protein complexes. And they are in the schemes associated with defined stages of the endocytic system, but in reality, these are proteins that are very difficult to localize by immunoelectromicroscopy, and we do not really know where exactly they are located, at what stages at what subdomain of the different endolysosomal organelles. And then, of course, we also have the lysosome dynamics. There's not one lysosome in the cell. There are hundreds of lysosomes, and they're not just sitting still, they're moving in the cell. And also that is very important for their function. So we have these three types of definitions of lysosomes, and we use them in a kind of parallel universes, because it is very difficult to connect these different definitions with each other. So we know about morphology, we know about functional proteins and about lysosome dynamics, but we know that from very different samples, and we don't know it from the same cell or from the same organelle. And that is what we would like to do, that we can combine all these parameters on the same organelle. And we do that by a CLEM. It was already introduced by Arnold. And by that, we make use of the advantages of light microscopy for protein localization and dynamic studies, and also the large field of view, so that you can very easily find your region of interest. We also make use of the advantages of electron microscopy, that we can see the ultrastructure, we can see the context, are they close to other organelles, and of course there are high resolution information. And what we do by CLEM is we combine those two methods on the same cell and the same organelle. And then we have CLEM, and by combining these parameters, we kind of introduce a new multimodal imaging tool. So I want to show you one example using Magic Red cathepsin B. And this is a marker that can localize cathepsin B, but you give it to the cells and it is membrane permeable, but it does not become fluorescent. Only it becomes fluorescent when it is cleaved by cathepsin B. So with an antibody, you localize all the enzymes. With this probe, you localize only the active enzymes. And this is, of course, also going back to the talk of Arnold. There's all these cool tools that you can now use in the live cells, but we cannot use them in electron microscopes because then you need to fix cells. So what we want to do is to combine that. And so this work was done by Nalan Liv and Job Fermi in the lab. And we asked a very simple question. So which of these compartments, the early endosomes, late endosomes, lysosomes, do actually contain the active form of cathepsin B? And well, this is just a movie. Oh, it doesn't work. Well, I'm a little bit, yeah. There. This is a movie of magic red cathepsin B, and you see that there is a lot of active lysosomes in these HeLa cells, and they are moving around so that you get an idea of how it looks in the live cells. And this is another cell that we incubated with magic red. Also, we incubated with dextran, which is an endocytic marker, which is taken up by endocytosis. We looked at the cells live, then we fixed the cells, and we made by the confocal microscopy a Z stack so that we can look at the different Z directions. And then we took this part of the cell and we imaged that by volume EM, and we used FIB-SEM. So this part you see now in FIB-SEM in 3 dimensions. We go from the top of the cell to the bottom. Here you see the plastic with some extensions. And now we are going back again. And now we overlay the fluorescent images over the organelles that we can find in the 3D EM image. So that means that in green, we see the organelles that only have taken up dextran. In red, we see the organelles that are positive for cathepsin D activity. And in yellow, we see the organelles that have both. This is a zoom in the same cell in which we selected 7 of these compartments in the light microscope. And this is the corresponding plane in the FIB-SEM image. It's a little bit tilted, so we miss number 7 here, but we can find back number 1 to 6. And for example, when we here look at number 5, it's green. It means it is reached by dextran, but it is not active for cathepsin B. And then when we look in the FIB-SEM image, we see this organelle, which is a little bit gray. And maybe you can see it, but it has a lot of intraluminal vesicles. And this is what we define as a late endosome. Now, when we look at this neighbor, we see it's much more black. And when we look at the fluorescence, we see it is shiny red. It means that it is very active for cathepsin B. Again, number 2. This is more an endosome, and this only has the dextran. And we also have organelles that have both dextran and cathepsin B activity. So we can do that for a lot of organelles, and we do that, we can do quantitations. And what we found is that when you do 2 hours of dextran uptake, that 94 of the late endosomes are reached by dextran and 73% of the lysosomes, which is understandable because lysosomes come later than late endosomes, and not all lysosomes might be reachable still by endocytic tracer. But what we found very interesting is when we looked at the magic red cathepsin B, only 23% of all the late endosomes were positive for magic red, whereas 84% of the lysosomes were positive for active cathepsin B. So what we conclude from that is that most of the endosomes lack the enzyme activity, whereas most of the lysosomes are active, at least for cathepsin B and at least for these conditions with this probe in these cells. I have to give that also as a disclaimer. But what we did see is that there's quite a jump in activity between late endosomes and lysosomes. So it does matter whether you as a cargo are in a late endosome or in a lysosome. And it does matter how the entry in the lysosomes is controlled. And that is the main topic of the lab that we are studying at the moment. So to get into a lysosome, we need the general membrane fusion machinery. Very shortly, we have a vesicle that wants to fuse with the target membrane. The first contact between the membranes is made by an activated Rab GTPase, which binds to a tether protein. The tether protein can make a conformational change, which brings the membranes into closer contact, and then the snare complexes can pair and induce fusion. So this is the very basic fusion machinery. And the Hobbs complex, which is the topic of study in our lab, is a tether. It is a multi-subunit tethering complex. And here you see the structure of the Hobbs complex as it is in yeast, which was by cryo-EM detected or discovered by the Ungerman lab. And here we see the model of Hobbs. It consists of 6 subunits. 4 of them, which are called the core, are shared with another complex, which is called Corvid, which I'm not going to talk about, although we do work on it. But there are 2 subunits, VPS41 and VPS39, which are really defining the role of the Hobbs complex. So there's quite a lot known already about the Hobbs complex, and many labs have contributed to the concept that the Hobbs complex mediates the fusion of late endosomes with lysosomes and also of autophagosomes with lysosomes, either directly or indirectly. So this is a pretty important step because, as you just saw, it can be the difference between being degraded or not degraded. And there are quite some patients with mutations in Hobbs subunits, and we have been looking at VPS41 a while ago. And the phenotype or the pathology of these patients is very severe. It is a neurodegenerative disease. They have a little bit of activity of VPS41 left, but still the transport of cargo from endosomes and the autophagy pathway to lysosomes is very much delayed in these patients. So the Hobbs complex really emerges as the point of no return, as the gatekeeper to get cargo proteins, yes or no, in the degradative lysosome. So this is well established, but it is not the entire story. Because Jan van der Beek in the lab, he wondered, so when you block this late step in lysosome biogenesis, what happens then upstream with the endolysosomal system? And here again, you see an EM picture of a wild type HeLa cell with this very modest lysosomes. And here you see an EM picture of HeLa cells that were knocked out for VPS41, one of the subunits of the Hobbs complex. And you see already that these cells contain structures that are much larger and much irregular in form than in the wild type cells. And what Jan found is that this Hobbs depletion causes enlarged endosomes, and these were positive for both early and late endosomal markers. Rab5, Rab7, LAMP, EA1, it's all on these compartments. They are not acidified, and they're also impaired in recycling of, for example, the mannose-6-phosphate receptor for lysosomal enzymes. So we concluded from that when you mess up with this late step in lysosome fusion, there's also a messing up of the endosomal system that precedes the lysosomes. And we were wondering how that can be explained. Yeah, and as I told you, lysosomes, of course, they have all these enzymes and membrane proteins, and those are made, most of them, in the endoplasmic reticulum. So to build a lysosome, you require the trafficking of more than 50 hydrolases and more than 100 membrane proteins. And we wondered whether the Hobbs complex was also involved in the trafficking of newly synthesized proteins to the lysosomes. And of course, that can be via exocytosis and endocytosis. But we also wondered whether the Hobbs could be involved in more direct and targeted and specialized pathways directly from the trans-Golgi network to the endosomes and the lysosomes. And we had a clue for that from previous work because in 2013 we identified a special type of carrier that came from the trans-Golgi network and that we called the LAMP carrier because they were positive for LAMP proteins, and they trafficked from the TGN to late endosomes. And these LAMP carriers were not clathrin-coated. They were devoid of mannose-6-phosphate receptor that is involved in the transport of lysosomal enzymes. And we found that they were transporting LAMP1 and LAMP2 to late endosomes. And interestingly, we found that they required the SNARFM7 and also VPS41 for this trafficking. And VPS41, again, is a component of the Hobbs complex. And we postulated at that time there are multiple pathways from the TGN for transport of lysosomal proteins. We have the well-established pathway via clathrin-coated vesicles and mannose-6-phosphate receptor for soluble proteins to the early endosomes. And we said, well, there is an additional pathway which we call the LAMP carrier pathway for lysosomal membrane proteins to late endosomes. So we wondered what could be the role of VPS41 in that pathway. And here is another zoom in of these carriers. So here we have a Golgi complex, and we nicely see the clathrin-coated vesicles that we know are transporting the mannose-6-phosphate receptor. They don't have LAMP. LAMP is in these small carriers that we also see here surrounding the late endosomes. So, and since VPS41 is part of a tethering complex and these LAMP carriers need to be transported to the late endosomes, we asked the question whether VPS41 is actually required for tethering of the LAMP carriers to late endosomes. And this work was done by Paolo Sanza in the lab. So what Paolo did, he took VPS41 and he added a mitochondrial targeting signal to that, TOM70. And normally VPS41 is of course found on lysosomes, but with this tag it is located to mitochondria. Where you can see that it here co-localizes with the mitochondrial marker TOM20. So this is not the real localization of VPS41, but we use this trick because then we can see what proteins are following VPS41 to this ectopic localization. And also we could see which proteins are following VPS41, but also the other subunits of the Hobbs complex. So we wondered whether VPS41 might be involved in the tethering of LAMP carriers. So here is here's VPS41 on the mitochondria, and here's LAMP. And we indeed found that when we express VPS41 in cells, we get an enrichment of LAMP-positive membranes on the mitochondria. And here you see the quantification of that. And also what was interesting, when we did the same with other components of the Hobbs complex, VPS18 or 39, we did not see that enrichment. So it was specifically VPS41 that could recruit LAMP-positive membranes. And we also looked at other proteins that could be recruited by VPS41. And surprisingly enough, it did not recruit its known interacting partner Rep7, but it massively recruited the small GTPase RL8B. Now, RL8B we know as a GTPase that is present on lysosomes, and via skip it can bind to kinesin 1, and it's involved in the anterograde movement of lysosomes. So we wondered, could RL8B also be required for the recruitment of the LAMP-positive membranes to mitofibrous 41? And we got cells from Juan Bonificino that were knocked out for RL8B. And then we did express again the mitofibrous 41. And now we found that LAMP was not recruited anymore to mitofibrous 41. Whereas when we overexpressed RL8B, we saw this massive recruitment of LAMP-positive membranes to mitofibrous 41 positive mitochondria. And this is the quantification of that. We also showed that when we deleted the RL8B binding domain in VPS41, that the LAMP vesicles were no longer recruited. And we showed that the mannose-6 phosphate receptor, which uses another exit from the TGN, is not recruited by VPS41. So, as I told you, RL8B is a small GTPase present on lysosomes. We see that it is recruited by mitofibrous 41 to the mitochondria. We don't know still yet, of course, what these LAMP-positive membranes are. They can be lysosomes, or could they be the LAMP carriers, or maybe both. So to find out what these membranes are, we did electron microscopy. And what you see here is a HeLa cell in which we knocked out VPS41. We expressed the mitoform of VPS41 and we co-expressed RL8B. And then we labeled for endogenous LAMP1. So here we see the Golgi complex, and we see there's quite some LAMP1 already in the TGN here. We see that the clathrin-coated vesicles are again nicely negative for LAMP1, but the LAMP carriers here are positive. And we also see that the LAMP-positive membranes that surround the mitochondria are small vesicles and not endosomes, lysosomes. Sometimes we find an endosome, but really not a lot. We did the same for LAMP2, and again we find that it's very small vesicles surrounding the mitochondria. And then, because we also transfected these cells with RL8B, we did a double labeling between LAMP and RL8B. And the small gold is LAMP1, and RL8B is the big gold. And we found that LAMP1 and RL8B, in fact, co-localized on these small vesicles. Now, and this is very nice because Arnoud already introduced these systems. What we then did is we combined the LAMP rush system with the FKBP-FRB system for VPS41. So basically we could induce the exit of LAMP from the ER and combine that with the recruitment of VPS41 to mitochondria. And when we look at normal rush LAMP2A, we see that LAMP is nicely going to all the lysosomes. But if we do that in the presence of the mitoform of VPS41, we see that the LAMP is not going to the lysosomes anymore, but is now recruited to the purple mitochondria. So just have to remove the thing. Yeah. To conclude these studies, what we find is that VPS41 as sole part of the Hobbs complex can recruit LAMP carriers to, well, in the normal condition, of course, to enter lysosomes. And what we think is that RL8B is required for the transport of these LAMP carriers and also for the interaction with VPS41. And the whole Hops complex is then required for the fusion of these LAMP carriers with the endosome lysosome. So this highlights a special role for VPS41 within the Hops complex, and it also highlights a role for RL8B, which was mostly known as a lysosomal GTPase, to be involved in the transport and tethering also of biosynthetic proteins. So how much time do I have left, Francesca?
We're a bit over time, but you can,
Okay.
if it's another minute, you can go, right?
Yeah, I will
I
go
guess you're the
now.
second one, so yeah.
I
Yeah.
have counted on that, so I just want
Perfect.
to I will skip this one, but I will go to the conclusions and show you that in addition to the LAMP1 proteins, we have shown, or we have now our data suggests that also the membrane component of the VATPase and also the cholesterol transporting protein MPC1 are transported by the LAMP carriers. And well, this I showed you already. There's also the transport of VPS41, RL8B, and FEM7. So it means these LAMP carriers transport membrane proteins. They transport the machinery that is required for the trafficking of these vesicles, but they also bring this machinery proteins to the endosomes, lysosomes. And I want to finish then with some more conceptual ideas or take-home messages. So I showed you that Hops is required for delivery of the endocytic, the autophagic, but also by synthetic proteins to endosomes. And what we see here is that Hops is required to modulate the cargo of lysosomes, but also links it to the acquisition of the proteins that are necessary for the functioning of lysosomes and also for the buildup of fusion machinery. And one of the concepts that we may want to discuss is that also cytosolic factors may use membranes for trafficking to specific destinations. And the final slide is that I think that are many more TGN pathways present than we know now. And that they can target to different stages of the endolysosomal system, which may help to carry out the many different functions of endosomes and lysosomes. And that brings me to my group. I already notified the people involved in the work. But I also want to thank my group, the whole team, not only the scientific, but also the support staff. I want to thank my collaborators and, of course, my funders. And I want to thank you for your attention and your patience.